So my lab is primarily interested in the basic molecular mechanisms by which steroid hormone receptors signal in the cytoplasm as well as nuclear. So prostate cancer, much like a lot of the other hormone-dependent cancers, relies on the hormone testosterone. I'm trying to see if I have to promote tumor growth. So this is a hormone-dependent cancer. Testosterone activation of the androgen receptor activates that receptor to go to the nucleus, bind to DNA, and elicit changes in gene expression that promote tumor growth. So the early stage treatments are anti-androgens that bind the hormone binding pocket. They're competitive antagonists. They prevent hormone from binding, and they're quite effective in early stage disease. But when this disease progresses to the hormone independent state or castration resistant state, those treatments are completely ineffective. But the key is, even in that castration resistant state, that receptor protein is still critical and a key target. And so if we can identify alternative ways to target this signaling pathway that don't rely on that hormone binding that pocket, then we may have good therapies for treating castration-resistant prostate cancer. So I've spent the better part of 2 decades of my life characterizing chaperone proteins that help this receptor, the androgen receptor, fold in the cytoplasm. And there's a wide variety of chaperone and co-chaperone proteins that are important for this receptor to fold to its hormone binding competent conformation, which we call the mature conformation. I won't tell this whole story, but we focused in on the FKBP proteins that are part of this mature complex years ago for one simple reason. Out of all of these chaperone and co-chaperone proteins, that class of proteins seems to be one of the few that shows specificity for small subsets of what we call HSP90 client proteins. HSP90 regulates lots of important things. FKBPs regulate only androgen receptor, progesterone receptor, and glucocorticoid receptor. Nothing else in the cell. And we know that because I made the knockout mice, and the knockout mice are androgen insensitive, progesterone insensitive, and cortisol insensitive, and that's it. So it looked like a good target for inhibiting androgen receptor activity in prostate cancer. This is the protein, FKBP52. We've done tons of work. This is the androgen receptor hormone binding domain. We've characterized that surface as the 52 regulatory surface. We've characterized this proline-rich loop, in particular this little groove up here, as being critical and likely an interaction surface with the receptor. And so those are the 2 sites that we would like to target, and we've targeted both. We have a drug called MJC13 we think targets this surface of the androgen receptor and prevents 52 from regulating. We've characterized a drug called GMC1 that binds this pocket. We think it reorients the proline-rich loop conformation and disrupts regulation of androgen receptor signaling. So we've got 2 different drugs working by way of 2 different mechanisms. This is MJC13. I won't focus on all the data. You can read about it. It was published in PNAS in 2011. But how the drug works is it binds to that receptor BF3 surface and causes that HSP90 chaperone complex to get stuck in the cytoplasm, and the receptor is prevented from going to the nucleus upon hormone binding, and so then it can't turn on genes and drive tumor growth and things. We've also used structure-based drug design to identify a hit molecule, we call it GMC1 here, that we think targets that PPI's pocket in the FKBP52 protein. We've shown that it also inhibits androgen-dependent gene expression and lots of other things as well. And we've also shown that GMC1 prevents receptor translocation of the nucleus upon hormone binding as well. So this is with hormone, more purple, more nuclear receptor. With drug, less nuclear receptor. So both of these drugs, and there's been a lot of data showing that 52 regulates receptor translocation and binding to the nuclear pore complex and import into the nucleus as well. So both of these drugs prevent that process. So at that time, that's where we were, and I was just like a lot of folks that you've all been mentioning in here. I've developed these molecules. Now what the heck do I do with them, right? So these molecules weren't very soluble in tissue culture, and I was just suggesting to Dr. J at lunch that we need to add a 5th box to Dr. Bennett's little classification system and just label it pain in the butt and put my molecules in that box, because they really were hard to work with for a long time, and we sat on them for a long time until I met Dr. J. So because of the formulation work that she's helped us with, we were able to move MJC13 finally into animals intravenously and look at effects on xenografts and show that it prevents tumor growth. GMC1 later developed. We moved it quicker because we had already met Dr. J at that time and we knew what the process was, right? So it moved quicker along. And so we were able to get both of these drugs into animals thanks to the core facility and the formulation work that they do, and at least making these soluble enough to get into animals. We also, because of that work and that animal data, we've been able to get sponsored research agreements and licensing agreements with Maya Biotechnology, which is actually based here in Houston and also in Chicago. So they've picked up both MJC13, GMC1, and they're working with us on the hit to lead optimization process and really working with chemists to do what you all have been talking about, which is really look at the chemistry and pick the right chemotypes, allowing us, we have molecules now that are more metabolically stable, more soluble, and that we can get composition of matter on as well, working with this company. So that's that screening process we've been working on them with. We probably got a new, based on GMC1, we probably got 6 new leads that are more metabolically stable that we can get composition of matter on that we're moving forward with. In the meantime, we've continued our large-scale in silico screens just to identify new chemotypes. We've got 4 or 5 new chemotypes that we're working on. Again, most molecules are going to fail at some point, so the more unique molecules working by way of this mechanism we can feed into the pipeline, the better. So we're working on that. And finally, our collaborators at Colorado have suggested that androgen receptor becomes more important in some subtypes of breast cancer. And based on that, we've been working to characterize our drugs in breast cancer models, and so we have submitted a bunch of provisional patents and things to cover them for use in breast, and we're moving in that direction as well. And so I'll end there. I'd like to thank Dr. Jay. She's been very instrumental in us being able to get that animal data and to secure these agreements with the company. So without that collaboration and that work with the core, we would not have been able to do this early on. So I thank you for that. And I will leave it at that. So thank you all.